Hello everybody, welcome back to Biology of Cells and Tissues, jumping into our immune system. So there's a lot of information in this PowerPoint, however we're not going to go super deep into all of it. A lot more of this unit, we kind of want to focus on kind of what is the the rough idea, the definitions, that kind of stuff, just to kind of understand it a little bit more. We're not going to do a deep dive into the immune system just yet. It's going to be a little bit more in the future. We'll start to see kind of a little bit of a deeper knowledge of that. As always, leave a like on the video, subscribe if you haven't already, as well as leave any comments if you have any questions about the material or ideas for future videos. So, starting with the immune system, we want to talk about two different types of immune systems that we have. I should say two different branches or arms of our immune system. So, first and foremost, immunity is the ability of the organism to respond to and repel invasions by foreign substances. Foreign substances can literally mean anything that's not supposed to be inside of our bodies. So, it can be from something as quote-unquote harmless as pollen or dust, or other particulates in the air that we're breathing. Or it can be more like, think about what happens like when you go brush up against like a rose bush or something and you have those thorns that try and poke you. Anything that's trying to get into your body that's not supposed to be in there, on a little bit more microscopic level, we have bacteria and stuff all around us. So if our bodies are built to protect us both inside and out from those things on the outside that are not supposed to be in. So the two kind of branches, we have an innate, also known as a non-specific immunity. Innate means built in. It means like your primary, your innate response. You'll talk about this a little bit when you get into like chiropractic philosophy, the body's innate ability to heal itself, meaning that the body does it by itself. We don't have to tell it to do any of those things. There's no additional prep work. It's just part of who we are. So our innate immunity, or that non-specific immunity, starts off with like our physical barriers. So think all of that skin, all those epithelial cells and stuff that we talked about that protect the outside of us, that protect all of us from those kind of things, from the things that we are not supposed to, that we don't want inside of our body, along with some secretions such as tears, sweat, mucus especially lining, the whole GI tract, lining the respiratory tract, lining the nose, all that kind of stuff. Then we have our acquired, also known as our adaptive or our specific immunity. Again, it's called this, backing up one sec, non-specific, because it doesn't pick out specific things. It tries to block everything that tries to come in. Versus our acquired or adaptive or specific immunity, it is built up and it is built as we live our lives. So we create this immune system by being exposed to different germs, as well as kind of through vaccinations, that kind of stuff as well. We build up immune system to very specific things in order to make sure that we are protected against them. So in order for our acquired immune system to work, we have a couple of different types of cells, or cells with different jobs, I should say. So we have cells that will present the foreign substance to the cells that are supposed to respond. We are going to have responding cells that produce antibodies. We should know by now what cells produce antibodies. Those are our plasma cells, and the plasma cells are derived from our B lymphocytes, as well as responders that destroy infected cells. So our infected cells, that's destroying them, it's going to be looking like our phagocytosis. So we'll get into that a little bit more. Here's just another picture again, showing those different things, the innate versus the adaptive going on there. You can break it down even a little bit more. Sometimes you'll hear humoral immunity and cellular immunity, which we talked about actually in the lymphatic system PowerPoint. We just briefly mentioned those. So understand that the humoral immunity Also known as the fluid immunity is the B cells, and then the cellular immunity is more of the T cells. Again, this is just kind of showing all of those different parts of our innate immune system that we need to remember, like the tiers. We in our nose, we have nasal hair cells. We also have mucous membranes, all that kind of stuff. The skin itself, the pH changes in the stomach, all sorts of that kind of stuff protect us from getting stuff inside of us that we don't want there. Now, kind of the combination of innate and, or I should say, the levels of protection between these two, we always say that our innate response, our innate, is our primary defense. So you can kind of think of it like if we have a castle, right, and we have a moat around the castle. Our first line of defense is that moat, and that is our innate response. Then after that, we have specific people guarding, we have guards, we have people patrolling, all that kind of stuff, protecting the castle. That second level, those people protecting, that is then our acquired response. And the more stuff that we see, the more specific those people guarding the wall tend to be. You have people who are specifically good at certain jobs. They recognize specific things or specific people, specific events going on, and then they're better able to respond to situations. Kind of an outdated example, but it's just a way to think about it. What it all boils down to, though, when we talk about these different immune systems, specifically the acquired immune response, it's all boils down to communication. How do all of these different cells communicate with one another in order to activate the immune system, in order to carry out what they need to do to protect us? A lot of this is done through cytokines. So within those lymphoid organs, those lymphatic organs that we talked about in the last one, and during any type of inflammation, the cells in our body are going to release these chemicals called cytokines in order to coordinate defensive measures. These are chemicals. They have what's called a paracrine mode of action. Paracrine means, para means next to. Crine is referring to kind of the endocrine type style of system where it releases chemicals. And so cytokines can react, they release chemicals. Specifically, they release, the cytokines are peptides and glycoproteins, and these go around and they interact with some of the other cells around them, and they activate parts of our innate and our adaptive immune response. Previously, at one point, we talked about chemotaxis. Cytokines cause ketotaxis. So it's kind of those words, cytokines, chemotaxis, that kind of all branch together. If you remember it, chemotaxis is the actual movement of our white blood cells, of our immune cells, to the area because they've been signaled by the cytokines. Okay. We're going to talk about antigens. A lot of this is it's a little bit difficult to organize this just because there's lots of moving parts. So, like I said, we're not focusing kind of on the whole flow of everything. We just want to know definitions and know each individual portion. So we talk about antigens. We've mentioned antigens before. These are the substances that can mobilize the immune response. These are the things that are going to cause our immune system to say, that's not supposed to be there. We need to do something about that. Okay? These can be soluble, meaning that they can just be free-floating in fluid, like in our blood or in our tissues, or they can be attached to cells. Especially think cells like bacteria cells, especially, can have these antigens. They can be proteins, they can be polysaccharides, or they can be lipids. Additionally, you can have complete antigens or haptens. A hapten, it's just an not a fully put together antigen, which means it just needs to combine with something larger, like a protein, in order for our body to have a response to it. Meaning that hapten is not fully developed enough for our body to care about it and for our body to say it's a threat and to attack it. Okay? We're going to learn in a little bit these words, these epitopes, aka antigenic determinants. We have a whole couple of slides talking about those just in a little bit. So we'll get there in a minute. When we also talk about antigens, there can be what are called self-antigens. Okay? So self-antigen are found on our normal cells. So we talk about, for example, I believe I mentioned this already in the previous video. We talked about our red blood cells and how part of our blood typing is that we have these specialized proteins on our blood cells which determine if we have our blood typing, if we have A blood type, B blood type, AB blood type, or O blood type. So if we have these proteins, let's say these are A proteins or A antigens, then we have A type blood type. Let's say instead, what if we have both A and B? Our blood type is AB. So these are antigens. So in any other situation, so let's say we just had type A blood, right? That's the way that we were born. And then somebody gives us a blood transplant, but they give us the wrong type of blood and they give us B blood. All of a sudden we have these antigens that we don't recognize. They are not self-antigens. And that stimulates our immune system, and we attack and destroy those wrong typed red blood cells because we think that it's an attack on our body from a foreign substance. You're going to get way deeper into blood typing and all that kind of stuff in physiology too. That's a little ways away. It's going to be more Tri3 kind of stuff as well. Okay? Anyways, continuing, one of the things that the antigens do is that they bind to the antibodies. Okay? The antibodies, these are Y-shaped proteins. I'm going to highlight a couple things that I want you to remember. They're Y-shaped proteins that find and attach to specific parts of the antigen called the epitope. There's that word again. And each antibody has unique tips that fit exactly with its matching antigens, like a lock and key. So think back to like biochemistry when we talk about enzymes and how enzymes specifically match up with their specific substrates. Same kind of idea here. We have the specific antibodies for the specific antigens. Once they are attached, the antibodies are going to tag the foreign substance. So it grabs onto that part of the antigen called the epitope, and then it tags it saying, hey, this is not supposed to be here. So then our body can activate the cells that are going to go and break it down and destroy it, so make sure it doesn't cause problems. Here's what our antibody looks like. These are those antigens or those foreign substances, whatever the part is that is called that is the epitope. It's the specific portion of the antigen that is recognized. Down here below, again, I have a full kind of breakdown of this process. You don't need to know it this deep. However, if you want to dive into it a little bit more, you can see it there as well. Okay, and that is actually how antibodies and antigens are going to bind. And we're going to circle back to this a little bit in just a little bit. Okay, so antigens often have several antigenic determinants, okay, also known as epitopes. Antigenic determinant, epitope, it means the exact same thing, okay? Most often, they have several different epitopes, so that way our actual immune system can respond to them in many different ways. Our antibodies can bind to them in several different ways. Each epitope can attract different antibodies, so that way our immune system works much more effectively in making sure that we get rid of everything. and it's going to make sure that we have a strong response, what's called a multi-angle attack against the invader. Know definitions, specifically know what the antigenic determinant is, know what an epitope is, know that they mean the same thing, and then know that there are multiple of them on each of that there are multiple types of epitopes on the structures that have that antigens, or on the antigens. This is just showing a little picture binding to all the different epitopes. So now talking just about antibodies specifically. We know that they're produced by the plasma cells from the B cells. They are glycoproteins, specifically from a group called the immunoglobulin. We'll see this abbreviation IG. So antibodies are glycoproteins, and specifically the type of glycoprotein that they are is an immunoglobulin. Okay? They're soluble, or they're attached to the surface of lymphocytes, and they recognize and bind to the specific epitopes. They are multimeric proteins. Remember, multimeric means kind of like that quaternary form. They're made up of 2 or more polypeptide chains. Specifically, they have 2 lighter chains and then 2 heavy chains. A lot of this is just kind of the specific anatomy, I guess you could say, of our antibodies. We have 2 regions. We have an FC region and we have a Fab region. Important things to remember are the bits and pieces here that are highlighted. So I don't want to go too deep into this. We'll see essentially the main thing to remember is that the Fab portion, the antigen binding Fab portion, is where the antigen binding site is, as well as the light chain. And then the heavy chain is the FC portion, and that's more of the constant region, okay? So we have a light chain, heavy chain. Both of these are both constant. And then we have a small variable region, and this is what makes the antigen binding site specific. Now, I broke this all apart just so it's a little bit more digestible. I'm not going to go through all of these. A lot of this is just kind of, hey, remember some of these tidbits. I personally would throw each of these little facts into like a Quizlet, just so that you could recognize some of the key points about them, and that is going to be enough, to get you through the exam. So you have IgM, IgG, IgE, IgA, and IgD antibodies. Okay, this is the picture. This has got all of them there, each of them individually. Important things to note. IgM is the first antibody produced in an initial immune response, and it's bound to B lymphocytes. IgG is the most abundant antibody in the blood. IgG is also, if I remember correctly, let me double check. Yeah, is the known as the passive immunity in newborns. So they have a bunch of IgG to kind of keep them going at the beginning. We have IgE. These bind to receptors on mast cells and basophils. If we remember, mast cells are a big part of mast cells, are part of our histamine reaction, which mostly has to do with allergies. Always associate IgE with allergies. IgA is located in secretions. And then IgD, again, mostly bound to B lymphocytes. So those parts that I've highlighted, those are like the key points. Again, you don't worry about memorizing all of this. It'll be a handful of questions on the exam, but these are gonna come back a lot over and over and over again in the future. So The more you can remember of them now, the better off you're going to be for the future. Okay, continuing. Cells of the adaptive immune system. We have lymphocytes. We've seen lymphocytes a lot already. Just going to touch on a couple of extra things. They have what are called cluster differentiation markers, also CD markers. This is the way that we identify and classify our different lymphocytes. How do we tell apart the different types of T cells from B cells? This is how we actually identify the differences between them. It's just a nice little picture. It's got a lot of information on it, but we're going to cover all of this in future slides. So starting with our B lymphocytes, here's the things we want to start associating. So on our resting or inactive B cells, the IgM antibody is bound to the cell surface with the Fab fragment facing outwards. Just like we see here, it's facing outwards, looking for antigens to bind to activate it. When the specific epitope binds to the IgM, the B cell activates and undergoes something called clonal expansion. Clonal expansion is just rapid proliferation of the activated B cell. So it divides rapidly, very, very quickly to give you a bunch of the cells so that it can find and fight all of the antigen that's going on in the body, the foreign substance. B lymphocytes, part of this is they differentiate into plasma cells and memory B cells. So part of that clonal expansion, it proliferates, it divides a bunch. Some of them become plasma cells to make the antibodies, and some become memory B cells. This is kind of the building up of our immune system. Kind of like, think about it like takes notes and then kind of stores and files those away, saying like, hey, this is what we saw. If this comes up again, this is how we're going to beat it. Which gives us what's called an anamnestic response. What anamnestic response means, that the first time we have an exposure to something, our body might, it realizes that something's going on, activates the immune system, and it fights it off. The second time we're exposed to that same thing, your body, as soon as it notices it, its response is much quicker and much more aggressive. And it really pumps out the antibodies right away to make sure that we don't get sick again. This is kind of like when you think about when you get certain diseases, or like when you have certain vaccines, we actually inject part of the disease organism into ourselves, whether it be like a live vaccine or a dead vaccine or whatever, which we'll talk about vaccines in in immunology course. It kind of primes the body. It gives us that mini first response. It gives us a very small first response. So that way, if we ever come in contact with those things again, we take advantage of the anamnestic response, and we have a much quicker, much larger response to protect us the next time we're exposed to something. This is just going over that. We have that primary response, and then we have the secondary response where we can immediately activate and get going very, very quickly. Here's that first exposure. There's that second exposure. Much more rapid, but quicker response. Primary response, secondary response. This can also be a bad thing when it comes to like allergies or autoimmune conditions. If you're exposed to something that you're allergic to, you can actually, if you're exposed to large amounts of it over and over and over again, each response can be worse and worse and worse. So that's what you got to be very careful with allergies and the way that those are managed and taken care of, depending on the degree of them. So that's our B lymphocytes. We also have T lymphocytes. They all have what are called T cell receptors, or T cell receptor, TCR, on the cell surface. The T lymphocytes only recognize epitopes which are bound to specific types of receptors, to receptor molecules that are on other cells. These are called major histocompatibility complexes. So T cells only respond to these MHCs. We're going to talk about those in the next slide. Also, just a quick little introduction. We have a couple different types of T cells. These are not all of the types of T cells. There's a bunch more, but these are the big important ones that we want to remember. And we have helper T cells. These are CD4. Remember CD, those are there that is, our cluster differentiation markers. Specific things the specific markers that we look at to determine, okay, what type of cell is this? So CD4 are our helper T cells. They are central to immune response and coordinate the response of the lymphocytes. We have two different CD8 markers. We have cytotoxic T cells and then T suppressor cells. The cytotoxic T cells are involved in the cell-mediated response. The suppressor cells actually suppress or down-regulate the immune response to make sure it's not too excessive of a reaction. Okay, MHCs, major histocompatibility complex. So these are integral membrane proteins, so they're found, within the membrane of cells. So on the surface of most cells that help the immune system recognize foreign molecules. These MHC receptors, their job is to combine with fragments of proteins known as peptides from viruses or other foreign substances and present them to the lymphocytes. Okay, so they find the bits and pieces. It's like, hey, these aren't supposed to be here. They grab onto them, and then they show it to the T cells and be like, yo, you got to do something about this. Please fix this. And that activates the T cells, and then the T cells can kick their butts into gear and do their job. Two different types. MHC class 1. Sometimes you'll see it written just as MHC1. Means the same thing, don't worry. These are found on pretty much all cell surfaces in the body, and they present antigens to the cytotoxic T cells. This is a little bit of extra information. The bullet points in black are the ones that you really want to know. In blue is just a little extra reading you can look at. And that's what our MHC1 looks like. Not too concerned about specifically the individual components of it. But you can see it's an integral protein, so it's part of the membrane of the different cells. MHC class 2, or MHC2, these are found on antigen-presenting cells, so more specific. Macrophages, dendritic cells, the activated T cells, also on B cells, and a couple of other ones. Specifically, these are the ones that you want to remember. Macrophages, dendritic cells, activated T cells, and B cells. Remember those guys. And they present the peptides, the protein bits, that they find to the helper T cells in order to coordinate immune response. So MH1, this is what we really want to know. MH1 talks to cytotoxic T cells. MH2 talks to helper T cells. Cool? Cool. We also have these other cells known as the antigen-presenting cells. We have macrophages, which deal with part of the ingestion and partial digestion of foreign substances. And they have the MHC2 on them. And then T helper cells, they recognize the MHC2 antigen complex, and they bind to the macrophage in order to go activate the cytotoxic T cells. So this is part of that MHC2 response. I would say it's under the umbrella of the MHC class 2, okay? Because macrophages, T helper cells are both involved within these. A couple more pictures. This is just kind of picture walking through it. I took the time and I broke down each of the different components a little bit more on down below. This is more of like a little bit of a summary. If you need more information on this, honestly, just read through this a couple of times. That's gonna be. You don't need to know every single little tiny detail and component of this. Essentially, just that what we talked about, where the antigen goes onto the macrophage, which produces that MHC complex to then activate the T helper cell, which then goes and they together activate the cytotoxic T cells. Next part, we're going to go a little bit over because we're almost done here. So the cellular immune response. This is just another slide talking a little bit more about the helper T cells. A couple key points, just the ways that they can ask more questions about this. So it plays a central role in adaptive immune response, activate both the humoral and cellular immune systems. Once it's primed by an APC, that's that antigen presenting, and the antigen presenting cells. The helper T cells do all of the following. They activate B cells and the T cells, induce proliferation, they secrete cytokines. The helper T cells essentially kickstart that immune system. One of the ways that they do that is through margination and diapedesis of white blood cells. We've talked about both of these things before, where again, we just need to know the definitions of these two specific words. Margination is attracting white blood cells that are in the circulatory system, specifically neutrophils, they essentially adhere, or they kind of get glued to the edge of the blood vessels. That is the endothelium of the blood vessels. And then they get stuck, roll along, they stick to it, and then they move through the endothelial cells. The moving through is known as diapedesis. So again, just know those two specific words, know the definition, and then we also have, don't know why this is actually, should probably be its own slide. We have our phagocytic cells, and hopefully by this point we know what phagocytosis is. The phagocyte cells, they're the ones that are actually going to kind of swallow up all of the foreign stuff, munch it down, digest it, break it all down, so that way it's no longer an issue. And this just goes through step by step, where the phagocytes adheres to the pathogens or the debris. They form a little vesicle pod around it. It lets in digestive enzymes that are carried by those lysosomes, and then it broken all down and destroyed, and then it removes the indigestible and extra material that it can't use, which is then cleared away as waste. This is a breakdown a little bit more detailed of that. I'm not going to go into it. You don't need to know each of these steps a bunch, but this is just a little bit more detail and kind of the names associated with those. And here's a lab picture showing this process happening, where we have a phagocytic white blood cell consuming a bacterium. And that right there is our immune system. Thank you so much for watching. Have a great day and good luck studying.